Record. Okay. So, good morning, everyone, and welcome to the webinar series organized by Therapeutic and Diagnostic System and Technology Technical Activity Committee of EMBS. And thank you so much for taking the time to join us. My name is Paola Saccomandi. I'm the chair of the Therapeutic and Diagnostic Systems and Technology Technical Committee, and I'll be your host for today's session. I'm really delighted to welcome Professor Stuart Campbell from Yale University. Professor Campbell will present his research activities, his brilliant research activities in the field of human engineering heart tissues for diagnostic and disease modeling applications. So, Professor Campbell, I will say a few words about you. Professor Campbell is a professor of biomedical engineering at Yale University. His laboratory conducts translational research at the intersection of different disciplines, including cardiac muscle physiology, multiscale computational modeling, tissue engineering, and clinical cardiology. He is the scientific founder of Propria LLC, a biotechnology company that uses human engineered heart tissue to accelerate development of cardiac therapies. And this will be the topic of the talk. Dr. Campbell received bachelor degree in bioengineering at Washington State University and the PhD at the University of California, San Diego. He joined the faculty at Yale in 2012 after postdoctoral studies at the University of Kentucky. Thank you, Professor Campbell, for being with us today. We look forward to listening to your talk. Before we dive in, a couple of quick notes. So this webinar will run for about 60 minutes. The session is recorded and a replay will be available afterwards on YouTube channel of EMBS. In the chat, I've put the link to the YouTube channel, and we will have a Q&A session at the end of Professor Campbell's talk, and there you can ask your questions directly to him. So today we are excited to explore Professor Campbell's field, so human engineering heart tissue and their capabilities for diagnosis and new therapies. Stuart, the floor is yours, and thank you so much.
Paula, thank you very much for the invitation and for that very kind introduction. It's great to be here and to speak to you today. These are my disclosures. My lab is focused not so much around a technology as really a set of human diseases known as cardiomyopathies. As you'll see, that has really led us to develop engineered heart tissue technology. But I want to say upfront that we got there by first being interested in this important set of conditions that affect human health. These are cadaver images of a normal heart. In cross-section, you can see the atria and the ventricles, including the interventricular septum. And this is a cadaver heart from someone with hypertrophic cardiomyopathy. And you can see that there's a vastly thickened interventricular septum and then hypertrophy or excess mass or muscle growth in the chamber walls of the ventricle. So one reason why this is an important disease to study is because it predisposes those who have it to sudden cardiac death. The LV hypertrophy also leads to exercise intolerance and greatly affects the quality of life because of that diastolic dysfunction. And when we look at explanted tissues from patients with hypertrophic cardiomyopathy, we also see that there's a high burden of interstitial fibrosis. The incidence of this in the general population is believed to be about 1 in 500 individuals, so it's not all that uncommon. And in the late 1980s and early 1990s, it was discovered that the autosomal dominant pattern of inheritance in families can be explained by these mutations to genes that encode proteins of the cardiac sarcomere. And the sarcomere is is the structure inside of muscle cells responsible for producing contractile force. So we begin with a mutation, and there's a phase where the acute effects of the mutation are acting on the tissue, but it's really a couple of decades um, until you actually start to see some of these symptoms and effects of that mutation that I've shown there on the right. And that's when you finally get to the clinical presentation. So my lab was interested in this period where the disease has not yet emerged, but the mutation effects are being felt. And we want to bridge that gap and understand what it takes to go from a normal heart to a diseased heart, and perhaps why it takes so many years. And to do that, we developed a sort of a new approach to creating in vitro tissues that could be studied. And we also use computational models, and I'll talk a little bit about that, but again, mainly focusing on engineered heart tissue. So I'm going to start by talking about this process of genotype to phenotype in hypertrophic cardiomyopathy, something that I will call the monogenic paradigm. One mutation leads to the disease. And as a corollary to that, I'll discuss what we call the VUS problem. That stands for variant of unknown significance. And I'll let you know what that is when we get to that point. And then I'll end the talk um, really with a discussion of diagnostics and what we can do as engineers and tissue engineers to move beyond this monogenic paradigm. Okay, so I've already showed you the hypertrophic heart. This is a schematic of the protein complex in muscle cells, just a part of the sarcomere, the thick filament and thin filament. As I mentioned, the inherited forms of hypertrophic cardiomyopathy are due to mutations to the proteins that you see represented here. But interestingly enough, different mutations to these same proteins can also produce a condition called dilated cardiomyopathy. Instead of the wall getting thicker, in dilated cardiomyopathy, the wall stays about the same thickness or maybe gets thinner, but the chamber dimensions get larger. And so that's kind of one of the interesting questions out there is what is it that makes a particular mutation lead to hypertrophy versus dilation? Can we understand and predict that? So there have been papers that sort of have worked on this problem, and they measured the twitch contraction, basically the shape of contractile force that's produced in a single contraction of the heart or of muscle isolated from the heart. And this is work by Jennifer Davis and Jeff Mulkinton, published now nearly a decade ago. But they advanced this hypothesis that if there is a mutation that increases the area under this isometric muscle contraction curve, that's going to lead to concentric hypertrophy. So the wall getting thicker and the chamber getting smaller. Whereas if the area under this curve gets smaller, so that's going down here, if that index gets smaller, then the tissue is weaker and the heart will dilate. You'll get what they call eccentric hypertrophy or dilation. Now, this work was done entirely in genetically engineered mouse models. We wanted to bring this into the human realm where the genetics, of course, are a little bit different. The heart rate is roughly 1/10 of what it is in a mouse. So we wanted to investigate this. Now, ideally, in order to do this, we would measure it directly in human tissue, but that, of course, is difficult to obtain samples, and you can't always get exactly the samples that you want. But when we began this work 13 years ago or so, this is when induced pluripotent stem cells were becoming increasingly available. And we teamed up with clinical collaborators to actually be able to collect blood samples and isolate T cells. And then we could induce pluripotency in those T cells and differentiate the cells into cardiac muscle cells or cardiomyocytes. But then the question is, how do you do mechanics? How do you do muscle mechanics on cells when they're just growing in a dish? So we worked on this problem and came up with a solution. which was to begin with a pig heart, cut a block out of the left ventricle, and then cryosection it into a thin sheet, making note of the orientation of the cardiac fibers. What we could then do is put this in a laser cutter and cut out a shape in which the cardiac fibers lie in the long axis of our roughly rectangular shape. And so now we have a macroscopically engineered shape, but with microscopic tissue properties. We decellularize that tissue to make a native collagen scaffold with aligned fibers. We generated a design for a tissue holder. We call this a cassette. And the tissue can be embedded in that cassette with everything that it needs to then have cells seeded into the construct. And this is a little video. Hopefully it shows up, but it produces, at the end of that process, autonomously contracting human-engineered strips. And these have a lot of advantages, including a native matrix. So the pig myocardial matrix is ideally suited for the growing and maturation of these relatively immature stem cell-derived cardiomyocytes. And the construct is also thin with aligned fibers, so we capture the anisotropy of the native tissue, and that also provides cues for maturation. So that's the basic technology. Another technology that came online during this time that has been very helpful, of course, is CRISPR-Cas9 for being able to manipulate the genome. So this means that we can either begin with a patient with a known clinical history and a disease-causing mutation, and then use CRISPR to correct that mutation. And then we have what we call an isogenic comparison. The genetics are the same between the two cell lines, except for the disease-causing gene. We can also use CRISPR-Cas9 to begin with a healthy cell line and introduce mutations of interest. And that's what we did here. So we looked at a gene called TPM1. TPM1 is the gene that encodes tropomyosin. As you will see, tropomyosin is kind of a gatekeeper for muscle contraction. It keeps the muscle switched off when it should be off, and then it moves out of the way to allow muscle contraction when the heart needs to contract. So this tropomyosin mutation E62Q, an amino acid substitution that is associated with patients with hypertrophic cardiomyopathy, HCM. We also looked at TPM1 E54K, which is a dilated cardiomyopathy associated mutation. And this pairing of these two mutations, the same gene, and in fact, they're only a few amino acids apart, but they cause dramatically different phenotypes in the heart. So the plan was we would begin with a healthy cell line. We would use CRISPR to homozygously insert these mutations, and then we could make the three groups of tissue and do biomechanical testing on them to find out what the differences were. So I'm going to show you that work here. Again, here's the paradigm, CRISPR engineer the respective mutations and then generate them, the engineered heart tissue. So here we can see an isometric contraction from the wild type engineered heart tissues. When we measure the same thing in the hypertrophic cardiomyopathy tissues, we see a dramatic increase in contractile force. So this is not what we would call a loss of function mutation, at least with reference to contraction. This is actually an excess of contractile activity. And then very beautifully, we see that with the dilated cardiomyopathy mutation, you see the opposite effect, that it drastically diminishes the contractile force. And you can see the summary data for that. These were very significant differences between the three groups. So hypertrophic cardiomyopathy seems to be associated with hypercontractility, too much contraction, and dilated seems to be associated with not enough contraction. Okay, so where do we go from here? We stretch these tissues to 3 different lengths. And if you have taken a physiology class, you have probably heard of the Frank-Starling relationship in the heart. So this is where when the heart fills with excess blood, the heart is going to contract harder to get rid of that blood. It's going to respond by increasing contractile strength. If we look at our wild type group, you see that behavior nicely displayed. Um, with a 10% stretch, you nearly double the contractile force. When we repeated this with the mutant tissues, the dilated cardiomyopathy tissues are already quite weak and don't display much of an increase in force. You may be tempted to say that the hypertrophic heart had a normal response to stretch because it has a similar slope, but they're beginning from drastically different places in terms of the tissue strength. So if we normalize each of these curves to the resting length tension, then we get the following. You can see that actually the wild type has the strongest length sensitivity, 2.5-fold increase in contractile strength with a 10% stretch, but neither of the disease groups show nearly that much sensitivity. So for different reasons, right? The dilated group, the stretch does not do very much to increase the contractile reserve. They simply can't recruit any additional strength. What's going on in the hypertrophic group is that they don't have as much reserve. Even at baseline, they're already producing almost their maximum contractile force. So stretch proportionally only recruits about an additional 50%. So this reflects a very drastic change in the underlying physiology and function of the muscle in both mutant cases, even though they have very different outcomes. So now we want to look at the what we might call the passive or the diastolic behavior of these tissues. So this could relate to how easily the heart is able to fill with blood in between beats. To measure this, we're stretching the tissue and keeping track of the baseline force as opposed to the peak contraction force. So we're looking at this sort of brown line and replotting this for each of the three tissue groups. You can see that the dilated mutation is making the tissue much softer and more compliant. And this actually makes sense when you look at the phenotype of the dilated chamber. There is less apparently that's resisting the stretching of the heart and may over many years allow the tissue to stretch out and to dilate the ventricular chamber. By contrast, you can see a much greater passive or diastolic stress in the hypertrophic mutant. And we wanted to know the basis of that. And so we reasoned that this could be due to hyperactive contraction even in between beats. This tissue may not be fully relaxing. So we treated it with mavacamten. Mavacamten is a myosin inhibitor that blocks contractility. And you can see that essentially normalized the elevated diastolic stiffness when we blocked that. So what's going on is this mutation is introducing a hyperactivity into the muscle, even when the muscle is supposed to be relaxed, it doesn't. Okay, so I'll take a sort of mechanistic detour here to talk about how we're using this system together with some models to understand why, on a biophysical or molecular level, these mutations are changing contractility. Cardiac muscle is regulated by the tropomyosin troponin complex, which I have schematically illustrated here. TPM1 or tropomyosin is shown in green, and as I mentioned, it blocks the binding sites for the molecular motor called myosin that produces contractile force. Now, what's interesting is that each of these spans along the actin filament is connected or coupled to each other at the ends so that we can think of the actin filament as being a series of coupled regulatory units. And this is just asking for some mathematical analysis. And what we do is we explicitly track the regulatory state of each one of these regulatory units along the thin filament. Each time you see a spike here, this would be a force-producing myosin actin crossbridge. And within each one of these regulatory units, we have a Markov network scheme with chemical kinetic rates that describe the activation of this system by calcium. Calcium ions are the second messenger in cardiac muscle that triggers the activation of contraction. And then we build in what we call nearest neighbor interactions, where tropomyosin, which is kind of represented by this bar, the tropomyosins are connected to each other. And so this generates a very interesting dynamic system. In 2021, we made this model even more complicated. We won't dwell on that here. But we focused on a couple of rates. One of them is the rate at which one of the calcium-sensitive troponin complex is resetting and binding to tropomyosin and actin. And I'll introduce one of the other thing that we're looking at here in a second. Again, I'll go quickly through this since it's not intended to be the focus of this presentation, but we had to create a mechanistic biochemical scheme to try and describe this. And we leveraged a classic model in the field, which thinks of these binding sites for myosin as either being blocked, where there's no myosin access, closed, where there's partial access, and then myosin bound M. And so we assumed an energy landscape for those three regulatory states. And we came up with an equilibrium constant for to describe the blocked and closed states, which really corresponds to movement of tropomyosin. Then we think of this tropomyosin chain as being approximated by a spring-like system where when all the tropomyosins are moved into a permissive state, that would be in the C state, there would be very little energetic penalty for doing that. But if the adjacent regulatory units are in different states, then that induces this kind of potential energy that would be related to the stiffness of the overall tropomyosin chain. And so that allowed us to write potential energy terms, which we could then use to superimpose onto our reference energy landscape. And now the spatial status of these proteins along the thin filament can impact the chemical equilibrium of the system. So that's the second parameter we want to look at, is the effective chain stiffness. So we have the equilibrium constant and the chain stiffness are two things that will go into this model. Okay, so the bottom line is that we can use this model to reproduce experimental data. And in the literature, the E62Q mutation was seen as shifting the calcium sensitivity of contraction. So if we look at how much contractile force is generated by the muscle for different levels of calcium, E62Q was seen as shifting that to the left. And E54K, it was seen as shifting that dramatically to the right. So it makes sense that we observed what we did. This muscle does not like to turn on after receiving this mutation, and this muscle is a little too eager to switch on. But we were able to recapitulate this with the model by changing specific parameters. And then we could simulate twitch contractions that are reminiscent of the ones that we measured in engineered heart tissue. And so that allowed us to conclude that E62Q, that mutation reduces tropomyosin stiffness and destabilizes that blocked state, because just by changing those two parameters, we were able to generate realistic model behavior. In contrast, E54K is enhancing the tropomyosin binding affinity for the cardiac troponin I mobile domain, which makes it difficult to switch the muscle on. So we produced these estimates from the model and looking at our data. The question is, if we looked at the molecular structure, would there be any evidence for this? And so we worked with collaborators at Boston University, and we actually did find some good evidence for this. They did a molecular dynamics simulation of the structure and found that there was, in fact, a 21% decrease in the chain stiffness of tropomyosin when you introduced that mutation. So that was a very interesting confirmatory result. Similarly, when they looked at the surface of actin filament and the location of this E62 residue, when that's mutated to, or sorry, when that's shifting between the blocked and closed state, you can see that E62 associates with K328 on actin, And when it's mutated to Q, that would be disrupted and thereby destabilize the blocked state. So that also seemed to match. We also noticed that the electrostatic interactions between tropomyosin and cardiac troponin I, oops, sorry about that, were increased significantly. Were decreased significantly by the E54K mutation. And so that seems to also align with what we saw with E54K. All right, so we wanted to look for a little bit more cellular evidence of how these mutations changed cardiomyocyte homeostasis. We cultured them in 2D and were interested to find that even without putting these cells into tissues, the area of the hypertrophic myocytes, E62Q, was substantially greater. And the aspect ratio, you can see these E54K cells are long and skinny, in contrast to the other two cell types, which had a normal aspect ratio. So that was striking. We went on to do RNA-seq and mass spectrometry to look at proteins. And we did find a common set of genes that were either upregulated or downregulated that coincided between the transcriptomic data and the proteomic data set. And so we were able to look at those and analyze them for common upstream factors. And interestingly, the hypertrophic mutation is activating this TEAD2 pathway. And in contrast, the dilated mutation activated a completely different program. What stuck out in terms of the pathways that were affected were mitochondrial dysfunction and a downregulation of oxidative phosphorylation in the hypertrophic case, and then wide-ranging effects on matrix reorganization, for instance, in the dilated case. So just to summarize, we were able to look at biophysical changes that were associated with these two mutations. We saw that in the hypertrophic case, it increased calcium sensitivity of the contractile apparatus, leading to elevated contractility. And basically the opposite occurred with the dilated mutation. And these triggered, in turn, recognizable and widespread changes in the biology of these cardiomyocytes, and then finally in changes to the cellular morphology that seem to match up with what is seen in patients. So what we've done is to build a much more detailed description of how these diseases develop. Now, there are pharmacological agents that could be used to counteract these. Mavacamten is the first FDA-approved medicine for treating hypertrophic cardiomyopathy for obstructive cardiomyopathy patients. And there's another compound that's being worked with called danicamtiv that's designed to increase contractility. And so we wondered if we could reverse the effects. These are both myosin-targeted drugs, but our question was, could we use a myosin-targeted drug to correct the errors introduced by a tropomyosin mutation? And so this is an illustration of how we can use these heart tissues to look at treatment. What we did is we incubated engineered heart tissues in the respective compound for 48 hours and then gave 24 hours of washout. Mavacamten is a crossbridge inhibitor, a myosin inhibitor. And as you can see in the wild type tissue, 48 hours of treatment with mavacamten did, in fact, kind of atrophy this tissue. It reduced its contractile force even 24 hours after washout. You can see once again the excessive contractile force in the E62Q tissues, and treatment with mavacamten brought that down into a normal range very nicely. So that's sort of confirming the biological hypothesis behind using crossbridge or myosin inhibition to treat hypertrophic cardiomyopathy and counteract hypercontractility. So the opposite is kind of true with danicamtiv. You can see that after a couple of days of incubating with this myosin activator, that the tissue, even the wild type tissue, comes back stronger. It's sort of increased the contractile protein content. And again, you can see very effectively this drug is enhancing contractility and bringing our formerly weak E54K tissues up to the wild type levels. So again, just a quick example of how we can test therapies using these engineered tissues. All right. So that kind of concludes this part, and maybe I'll ask the question, so what do we learn? Let's now go to a little bit more of the clinical scenario to understand why these discoveries are relevant. If you look at all the patients who come in of every kind with hypertrophic cardiomyopathy, there's a large percentage of them, about 40%, that have no family history, and they don't have a mutation that's recognizable. About a third of them do have a recognizable mutation that's pathogenic or likely pathogenic, and that's helpful, okay, because if you have a patient who comes in, they've maybe been experiencing chest pain or had an unexplained fainting episode, something like that, you now immediately want to know whether there's family members who are at risk. Everyone in this particular family tree with an arrow is somebody who should be investigated to make sure that that they don't also have hypertrophic cardiomyopathy, have this risk for sudden death, et cetera. So with genetic testing, in this case, the patient was found to have a mutation in myosin heavy chain, MYH7. And because that was a known mutation that had already been linked with hypertrophic cardiomyopathy, they could diagnose this sibling with hypertrophic cardiomyopathy and refer them to clinical care. They could find that the sister does not have the mutation, and so neither she nor her children are at risk. However, among the two children of this initial patient, one of them carries the mutation and should be seen by a physician every year or two going forward. So this is what we call risk stratification through cascade screening, and it is very helpful. But the problem is that for many patients, their variants are not known, and so you can't do this type of screening. So we're interested in this piece of the pie, these 9% of patients that are living in some kind of limbo because their family has the presence of this disease. There is a suspicious mutation, but physicians cannot treat or refer members of this family on the basis of a VUS, a variant of unknown significance. So how can we classify these variants, and how can we do it in an accurate and scalable way? My group has done work with this family in particular, a different mutation that causes another form of cardiomyopathy. In this case, we took several years to genotype 100 different people in this family. It was a lot of work. It was successful in changing the classification of this variant, but it's not scalable. We can't do this for every single family. We could use an animal model, but animal models are also not very scalable, and they are also not very accurate in many cases. So we could use some of these bioinformatic algorithms. In fairness, this polyphen2 algorithm is now many years old, but just to test it, we plugged in random variations in TPM1 into this model, and we see that about half of them are classified as benign and about half as pathogenic. Then we put in a set of known HCM mutations in TPM1, and it turns out that they're distributed basically throughout that entire range. So that's not very helpful. So what can we do? Can we do this better? We've been working on a pipeline to do this now with our collaborators in Boston University, trying to use atomistic models and simulations to predict some of these parameters that I introduced to you earlier and see if we could plug those into the model and then predict in vitro motility assays and twitch contractions and then validate them experimentally. And we are making some progress on that front. We've published in that area recently as well. So that's promising, and we're going to keep working on that. But we actually determined that there might be an intermediate throughput method just focusing on engineered heart tissues, and I'll tell you about that really quickly. So what we're doing is we can actually generate a normal, healthy engineered heart tissue And we can incubate it with an adenovirus that expresses a tropomyosin variant of interest. And that whole process from uh, generating and culturing the tissues and then growing them for a week with the virus, that process takes about 3 weeks. And we've been able to demonstrate that expressing an exogenous tropomyosin overwhelms the natural or endogenous tropomyosin, achieving about a 50%, 40 to 50% replacement of the normal protein. So what we did is we took variants of unknown significance from the clinical databases, and we engineered these viruses that contained those variants in tropomyosin. And then we also, for benchmark, you can see our old friends, E62Q and E54K, as our hypertrophic and dilated benchmarks. And then we also use wild type tropomyosin as a control in each case. And this actually seems to work pretty well. So what we do is we generate a batch of tissues and we randomly assign them to either receive the wild type virus or the virus that contains a mutant tropomyosin. And then we can measure their contractile function. And for this variant A102D, we noticed that there was a consistent and significant increase in the twitch contraction force and a significant delay in relaxation. So this looks a lot like our hypertrophic phenotype. We repeated this for D258E. This time we just picked up a subtle difference in the relaxation time, even though the forces were about the same. A239T mutation nearly completely abolished the contractile force. And K233N also had a similar behavior, reducing the contraction of these engineered heart tissues. So we wanted to compare this to a different assay to see how we were doing. Our colleagues at the University of Massachusetts Lowell, Jeff Moore's lab, performed what they call an in vitro motility assay, looking at how actin moves along a bed of myosins from purified expressed proteins. And we saw a really good agreement. You can see that there's more myosin activity at the same calcium level, so an enhancement of calcium activation or calcium sensitivity for A102D. And a slightly subtler phenotype, but still you can see that at low calcium, this D258E does not like to switch off, which may explain the poor relaxation rate. And then we get rightward shifts in these curves showing a drop in calcium sensitivity in both of these mutations that seems to track well with what we observed. So overall, this seems like an efficient way to characterize these mutations. We can characterize a mutation using this method really within about 2 months. Okay, now just for a quick interpretation, we go back to this idea of looking at the area under the curve as an index for what the phenotype will be. We call this RFTI, and you can see that for a normal tissue, it should be just below 0.2. If it's above this line, that's too much contraction. If it's below, it's not enough. And we see here's our HCM benchmark is above the line as we expect. Our DCM benchmark is below the line. And then we can look at each of these 4 variants, and you can see that they classify pretty nicely. This one would be trending towards excess contraction, but did not quite meet that mark. So that may be a less potent mutation. All right. So we predict that they fall HCM and DCM in those ways. I think in the interest of time, I'm going to skip over the slide. It's just repeating some of these tests in 2D. So we think, just to summarize our work, it's very challenging to go from genotype to predicting the phenotype, rather than saying that we'll try to do that for every possible type of mutation in muscle. We've picked a very specific path up this mountain, and that is to look at mutations in tropomyosin. And our computational models are gaining some traction, but we have to keep working on those. We're seeing that we can already do some direct genotype phenotype predictions using these tissues, so we'll continue to work on that. In the last part of my talk, I briefly want to tell you about the more difficult parts of this pie. Okay, I've already told you that there's some very useful clinical things that we can do over here on this side of the pie. And we've also been able to show that these correspond with contractile changes in engineered heart tissues. But what about over here where there's not a clear genetic link at all? Can we do anything for these patients? And we think that we can. We believe that we can create engineered heart tissues from patients in this group and look for some of the similar changes that we saw on this other side of the pie. And so that's what I want to tell you about. So what we did is we recruited and developed cell lines for 7 different genotype-negative patients. So they're from that part of the pie. And then we put in for positive controls a couple of genotype-positive patients. We blinded ourselves to the status, created their IPS lines, and then made engineered heart tissues to look at their contractile behavior. Then what we did is we developed a panel of 11 healthy reference individuals so that we could have kind of a bank of normal contractile behavior. And it turned out that this number was higher than we had initially believed, as I'll show you in a minute. But we had to have this rather large reference panel. To do this, we actually ended up using the engineered heart tissue system that was developed by the commercialized version of this technology, Propria LLC. From these tissues, we can get things like the Frank-Starling slope, the length-dependent activation slope. We can get the relaxation time, the peak force, the area under the curve, and then the ratio of the contraction speed or contraction rate versus the relaxation rate. So these are 5 metrics that we wanted to use to see if they were changing across these different patient groups. Now, what's interesting is that, as I mentioned, there's a lot of natural variation in the behavior of tissues generated from healthy individuals even. And we noticed in particular, this cell line, S5H, was from a healthy volunteer, and it had the lowest length-dependent activation, the least sensitivity to stretch of anyone. And so we actually had to run whole exome sequencing to look at the genetics. And we actually found that this person, unbeknownst to them, carried a variant of unknown significance in FHOD3. And when they were referred, so we went back to this volunteer and suggested that they get an echocardiogram. And they did, in fact, show mild septal hypertrophy. So even before we got this study started, we accidentally provided a diagnosis for a volunteer in our cohort. So moving forward, we actually excluded this person from that cell line from the reference panel. And we now have a reference panel mean that we can compare our cell lines to. Every time you see a star, that's because it's statistically different from the reference panel mean. And not all of our genotype negative patients showed a difference from the mean, but one of them actually showed an excess length sensitivity. And you can see that these others showed a diminished length sensitivity. I guess there were 2 that were marginally above the line. You can see these in orange are our sarcomere mutation positive patients that would have already been caught with genetic testing. But you can see that there are some other individuals that who would have been revealed through this direct engineered heart tissue type diagnosis. And when we expand this and look at all 5 of these parameters, you can see that there's varying degrees of significance in the different metrics. Our genotype positive patients, these are our positive controls, they had very large septum. So normal septum should be below 1 centimeter, and these were both nearly triple that. And you can see that they differed in most of these contractile parameters. And then we get a range of responses in our genotype negative patients. There are several of them that have significant differences, and then there are some that have only very, occasionally will show a difference or maybe even no difference at all. I will note that these tended to be the patients that had more mild hypertrophy. So just in summary, we set out to see if patients from this group might have different behavior in their engineered heart tissues, and we did indeed find that in some cases they do. And the conclusion that we draw is that it could be that contractile abnormalities are playing a role in this sort of mysterious part of the patient population. And so we do think that there is some potential for using patient specific engineered heart tissues to assess risk in patients. Clearly, a lot more work to do. I just want to conclude by thanking our sources of funding, primarily the National Institutes of Health. I also want to thank the wonderful members of my lab who contributed so directly to this work. The tropomyosin story that I told you at the beginning was the work of Jeanette Creso and Shwethi Halder, who have since graduated. They were supported tirelessly by our lab manager, Xiao Li, who cultures the cardiomyocytes and assists in the work. And then these were undergraduate students, Yusuf Rashid and Lynn Kim, who have since graduated and moved on as well. But they, along with Shwethi Halder, really pushed forward our patient-specific genotype-negative modeling work. And then finally, our tropomyosin collaborators at UMass Lowell in the lab of Jeffrey Moore, and then Mike Rinkowitz and Bill Lehman at Boston University. So with that, I'm happy to take any questions.
Thank you, Stuart. It's fascinating. It's fascinating. Any question from the floor, please? You can raise your hand. Meanwhile, I'll start with one question.
Sure.
That's very far from what I know, what I do, so might be a silly question.
That's fine.
Now it's really wonderful. How many patients? So for building the reference panel you show us in the last slides, how many
Yep.
patients you needed, or are
Yeah.
you still recruiting? How does it work?
Yeah, that's a good question. So there are um, publicly available banks of induced pluripotent stem cell lines. And so some of those reference panels were procured by going to those biobanks, and then academically for a small fee, you can obtain those cell lines. We don't know much about those patients except that they're free from as far as they know, they don't have cardiac symptoms. We also recruited a few additional controls from our own program here. So just volunteers who are willing to give a blood sample. Um, so that's what we ended up doing. And as I said, we started with 11 and ended up doing genotype screening on those and eliminated one of them for the reasons that I mentioned. Um, so the reason why you need that many is just because of natural variation. And I think that by the time we got to 10, those reference numbers were stable and I think predictable. But it turns out that there's quite a bit of natural variation even in cardiac dimensions between individuals. Some people just naturally have a larger or smaller heart, which does not pass the threshold of being clinically problematic, right? And so I think we had to have that many to kind of average out some of the variation. But if we, tried this, I think initially we were hoping to use just one male and one female control, and that's not even close to being able to um, account for the variation.
Yeah, and this exactly what you said leads to my second question. Have you experienced or noticed some difference between male and female?
Uh, yes, yeah, great question. We looked at that in the reference panel, and there isn't, in
There is.
fact, a significant difference between the two. So the variation between individuals exceeds the variation between male and female.
Between gender. Okay.
Mhm.
Oh, wow.
Yeah.
Yeah.
So plenty of work left to do, but
Yeah.
first
You,
things.
you said 13 years of research, right?
Yeah, that's right. So my lab was started 13 years ago here, and in the beginning, it's just developing the platform and making sure that it works and validating it. And But since then, we've used it for a lot of different applications, and we continue to find uses. I think one of the more exciting things we're doing now is to create devices that can administer different mechanical loads to the tissues while they're growing. Instead of just doing the biomechanical testing at the end,
Okay.
we can use mechanical inputs as an experimental variable, and change the loading to reflect lifestyle differences, exercise, that type of thing. So there's, as I said, plenty of work to be done there to access new questions that have to do with mechanotransduction. Mhm.
Uh, and I have a curiosity. You at the very beginning of your talk, you showed the video of the contraction of the of your engineered tissue, her
Yeah.
tissue. Um, so how for how long those probes, I don't know if probe is the right word, can keep contracting?
Yeah, that's a great question. So we're attempting to emulate ventricular uh, muscle cells, cardiomyocytes, and ventricular cells are normally don't beat on their own. Um, as we grow these tissues longer, their intrinsic beating rate actually slows down, so it becomes more mature. So when we want to measure these tissues, we electrically stimulate them at the rate that we would like. Um, but to answer your question, they start beating within about 24 hours of forming the tissue in the scaffold, and then we can keep them alive for many months, actually, and they're just fine. One of my students actually carried one out to a year. He didn't ask me if it was okay to use all of that cell media, but he did it and it stayed alive. So
Wow. Stuart, we have a question from Haritz. Haritz, you can open your microphone and ask your question.
Okay. Hello, can you hear me?
Yes.
yes,
Yes, a good lecture, by the way, Professor Stuart Campbell. So, I actually I'm actually a PhD student, currently in the field of cardiac tissue engineering, creating a bioscaffold and stuff, right? So,
right.
first off, I'm actually very impressed with the work of the TMP1 genes, which you actually induced on the healthy cell lines, correct? The healthy cell lines of the cardiac cells, right?
Right.
So, for that reason, the one that you showed the metric showing that the contractility is actually better. Is it comparing with the HCM or DCM? Or is it actually comparing with the healthy cell lines patients? The metric line, I think it's a Frank-Starling graph, is
Right.
it? Yeah.
Yeah. So that's a good question. It turns out that I think of this in terms of homeostasis, right? So the heart has a normal size and shape, um, that is associated with somebody who will live a long life and will not have problems with their heart. What we're really learning from this HCM and DCM story is that it does not matter whether you have too much contraction or if you have too little contraction. Eventually, over many years, that's going to pose a problem. That's going to push the heart outside of what it can homeostatically compensate for. Now, there's a lot of compensation, but it takes time. So I don't know if this answers your question, but we can't really say, is this mutation making the tissue better? Um, from a strictly just looking at how much contractile force is produced, the HCM mutations do cause the tissue to be stronger. So if you're trying to engineer a stronger tissue and you don't mind losing the diastolic behavior, the ability of the tissue to relax efficiently, then yeah, I guess in that sense, the mutation may make the tissue better, but that doesn't necessarily mean that it's a good thing. In fact, inside of a patient, it winds up being a bad thing. So I don't know if I answered your question though.
Yes, yes, yes. Thank you very much, Professor Stewart. So it's actually interesting where you discover that it can eventually improve to support the against the HCM patients, right? You're improving the contractility. That's what from I understand from my side.
Well,
But Professor Stewart, like if for example, do you actually happen to estimate how long can you sustain this improved contractility strength? Like, do you estimate how many years if we actually implant these EHD cells into the HCM patients?
Oh, I see. Like using this as a form of regenerative medicine, like implanting tissues. Yeah, I mean, I think that that's a good question. We have not specifically worked on developing tissues for implantation um, to help the failing heart, right? Whether that's due to myocardial infarction or non-ischemic cardiomyopathy or non-ischemic heart failure. But we think that, there definitely are lessons that could be learned from what we're doing to apply to those applications. And there are a lot of uh, groups that are doing wonderful work in this area of how do you form and mature units of engineered heart tissue? How do you vascularize them so that you could put them into the failing or the damaged heart? So again, although we're not doing that work, I do think it's very interesting and a lot of exciting stuff being done in that realm.
Thank you very much, Prof.
Thank you for the questions.
Yes, I see another question. Andrea, yes?
Yes, thank you so much for the presentation. I was curious, can you do this very same procedure to mimic different tissues, or is this just limited to heart tissue because of the fibrous structure?
Right.
Do you think this can be replicated for different organs?
Yeah, that's a great question. Being engineers, we started with the goal of making the best mechanical evaluation of these tissues. And so I think that this is well suited for other applications for tissues where mechanics are important. So we actually worked with Laura Nicholson's group to create decellularized slices for lung tissue engineering. And in that case, of course, you don't necessarily think of the lung as a mechanical organ, but with respiration, it's under this constant load that's oscillatory. And so that allowed us to create a device that would subject the tissue to periodic cycles of stretch and relaxation. And that was successful. So we have moved into that space a little bit. I think other types of muscle, smooth muscle in particular, and skeletal muscle would be very interesting. Growing skeletal muscle in terms of the cells can be challenging, but there are many groups now that are beginning to do that. So I think skeletal and smooth would be a natural way to expand this technology. And then I guess also thinking of skin could be another area where mechanics are important and where it might be feasible to create those cultures. So I hope over time that we'll expand into there.
May I just make a follow-up question? So, how big is the biggest sample or the biggest tissue that you have grown so far?
Yeah, so with one of my colleagues, Yibing Chang, here at Yale, we've actually created some that are on the scale of, maybe a little bit over a centimeter square. And then they can be used for regenerative type applications. So we haven't done a lot of this, but we have explored that. So if you can imagine maybe 10 millimeters by 10 millimeters.
Fantastic. Thank you so much. This is really amazing job. Thank you so much.
Thank you very much.
Okay. Yeah, I don't see other hands on. We are perfectly on time. That's great as well. So, Stuart, I would really like to thank you again for joining us for your great talk. It's amazing for me. Your research field it's like a frontier or something that could have come in few years, but no, it's now because you are doing it. I wish you all the best. It's really important. I really see the benefits of yeah of your research for patients. So or for everybody actually, for everyone. So best of luck.
Thank you very much. Thanks again for the invitation. I've really enjoyed it. So
Thank you. We enjoyed a lot as well. Yeah, now you are free. Let me thank also the all the attendees. And as I said at the beginning, this session has been recorded, and you will see Professor Campbell again on YouTube.
okay. Yeah, thanks again.
Thank you. Have a great day. Thank
Thanks.
you all. Bye bye. Bye bye.
Bye.